Okay, thank you. Hi everyone, my name is Yuqi Cheng, and I'm a second-year PhD student in Professor Xu Weizhang's lab at Georgia Tech. Okay, I will first provide a brief introduction to the problem we try to solve, and then I will show our method and some results, and finally, discussion section is at the end of the slide. Okay, let's start from the background. In single-cell ATAC-seq analysis, cell type annotation is the most fundamental step before any downstream tasks. We usually have two annotation approaches. One is to label cell types manually by marker genes. This is straightforward, but it's not very suitable to single-cell ATAC-seq data because peak information is not directly related to cell type. And we need to translate it into gene score. But sometimes the gene score can be inaccurate, this misleads annotation. Another way is to transfer cell types from constructed reference atlas, which is also called reference mapping. I believe we all get very familiar with reference mapping, which can be much more faster and more generalizable than the manual labeling. And the good news is that there are many single-cell ATAC-seq data atlases have been published in recent years. We can find over 2.5 million label data for human and over 1 million label data for mice. However, actually, we will also find that these data sets are not fully utilized, at least until now. So the first reason is that features are distinct between reference and query datasets. But we know that current reference mapping tools and supervised classifiers always require aligned features. But finding intersect peaks will cause information loss, especially in the scenario where the reference dataset and the query dataset comes from different reference genomes. And binding the whole genome can provide such aligned features, but you may say that this will produce median level feature dimension, and this will make the training of atlas level classifier is not practical. And only using gene score sounds like the most reasonable one, since it can provide a unified feature space with a limit dimension. But the gene score is not always accurate. Taken together, there does not have a good method to deal with this problem. And meanwhile, current single-cell ATAC-seq annotation tools cannot utilize multi-source references because of the previously stated feature problem and potential batch effect. And meanwhile, these existing methods cannot detect out-of-reference cell types in the new data. So this further obstacles the utilization of these methods in real biomedical research. So here, we propose our method, APAC, which is a heterogeneous transfer learning framework that models single-cell ATAC-seq data with distinct peak features and integrates later embedding in the common gene space. The first step is reference construction. Here, we use bridge integration to perform heterogeneous domain adaptation. We first train a sample-specific autoencoder for each reference sample to get low-dimension representation. And then the bridge variation autoencoder takes the gene score as an input. Each cell in the gene score embedding is constrained by its own corresponding peak embedding. And actually, we will say that the autoencoder structure here doesn't play a very important role. So we are trying to remove this autoencoder to say if we can improve the grading flow of this model. And after we integrate peak embedding by the peak line loss, the MMD loss will also be applied to them to bridge the gaps between the batches. So by doing so, we can partially transform gene score information to peak information while using a aligned feature set. And And with such a gene feature set, we can then perform transfer learning with pre-trained bridge VAE model to map query data into the reference space. And by fine-tuning the network, we can obtain the common latent space. Thus, we can easily perform cell type annotation on it. Here, we use a plain key and classifier to, sorry. Here, we use a plain key and classifier to transfer cell label. And after annotating the query data, we use a Mahalanobis distance to examine whether the label is confident enough. Here, since our goal is to detect major discrete out-of-reference cell types, a distance-based method can be used in this linear space. And we can calculate the Mahalanobis distance based on the statistical parameters of the cell type in the reference data, and then calculate the distance for each query cell based on the previously KNN result. And we can easily find that this distance obeys a chi distribution. Thus, we can perform significance test based on the distribution function and then obtain the rejection threshold. And query cell with an unconfident label will be recognized as unknown cell. So now we have the whole framework of ATAC-seq, and we can see some results. We first benchmarked the de novo reference atlas construction ability of ATAC-seq because we know that this is the very first step before we performing transfer learning. And we benchmark this ability against other popular batch integration methods. In this task, we test method on the dataset with the same reference genome. And all other methods are tested on the aligned peaks. And we can find that ATAC-seq outperforms the other methods on multi-byte single-cell or ATAC-seq data integration task, and compare with Harmony, Lager, or other integration methods. And on both small-scale mouse brain data integration task and large-scale Hillman PBMC data integration task. And UMAP visualization also verifies that the common latent space is reliable. And these results suggest that ATAC-seq is able to produce a reliable common latent space for reference mapping. And we know that the common latent space is a base for successfully performing the transfer learning on the query data. So this can provide us a solid foundation, and we can move to the next step. Before the next step, we also notice that sometimes the potential reference sources do not have the same reference genomes. So we also test ATAC-seq on this scenario because we don't need aligned feature peak, so peak set. So this may also one of our functions of ATAC-seq. So we benchmark this function. The other methods are tested on the remapped aligned peak site. And we also added 3 gene scores-based integration methods, Harmony, Lager, and ICVI. And the results show that ATAC-seq keeps outperforming the other methods in this task. And meanwhile, ATAC-seq also shows a good capability to keep small cell cluster, such as dendritic cell in PBMC dataset. I circled it in pink. Let's see, in here is a. And now we can move to the reference mapping task. And we then test whether ATAC-seq can successfully map new data with pre-trained model. So in this benchmarking test, we used 20,000 PBMC cells as a reference and 10,000 PBMC cells as a query. Compare with the gene-based ICVI plus ICRTs and aligned peak-based PIC-VI plus ICRTs. Our method shows better performance on the single-cell ATAC-seq transfer learning. And meanwhile, we can also find that ATAC-seq could also obtain a similar result as a de novo integration. The table 2 shows a comparison between the transfer learning of ATAC-seq and de novo integration of ATAC-seq. And this result shows that the design of APAC provides a model of better capability for single-cell ATAC-seq reference mapping. And if we got successfully map new query data to the common latent space, we can then transfer cell type. And the visualization result also shows that APAC is able to keep rare cell type in the reference mapping, such as PDC cells in PPMC dataset in query data only mode. And as the circle in blue, we can find the PDC only located in the query data, and we successfully map it separate from other major cell types. So on this common latent space, we can perform cell type annotation, and we can say that the APAC cell type annotation function also performs the other method designed for both like single cell ATAC-seq data or single cell RNA-seq data. And the Mahalanobis-based OR detection method also performs the current popular used KNN detection method and SVM detection method. So this result further strengthens cell type annotation function of APAC compared with the other popular used method. We use the F1 score as a cell type annotation matrix, and AUROC and true positive rate as a matrix to quantify the order of reference detection performance of APAC. And here is a case study of the APAC. We can see that the query data only contains the B cells. We can see this. We can see this as I circle it in blue, and this B cell is not located in the reference data set. And by applying the annotation and OR detection, we can see that the B cell successfully labeled as unknown cell type in the predicted cell type annotation result in the figure in the right first panel. And in the right down panel, we can see the APAC could also provide a certainty score because we have a probability function, and then this comes from the cat distribution probability, and we can see the unknown cell type also is a query specific B cell type is labeled as actually there approximate 0 probability. So this can be provide an interpretable result on the cell type annotation. And yeah, this it's the last slide. So in our future work, we plan to apply APAC to rebuild reference atlases for all human tissues and provide a ready-to-use pre-trained model for users to map new data sets to annotate cell types in their own data set. And APAC will also provide a comprehensive single-cell ATAC-seq data analysis platform based on Python, and we will try to release it in 4 to 5 months. And we will also keep improving the gradient flow to see if we can make it more lighter and faster, and perhaps we will try it on spatial ATAC-seq data if possible. And this is our future plan, and we will complete all these tasks in near future. And I would like to acknowledge all my research group mates for their support to my work, and the PI, the Professor Xiu Zhang, and the other PhD student in my group. And that's all for today. Thanks so much for your listening. Thanks so much.
Questions for you, Chief? Go ahead.
Yeah, sure.
So, given a new, sorry if you may have missed it as part of your talk, but if you have a new single cell ATAC-seq data, do you have to retrain APAC, or can you use it somehow map the new data set onto the latent space in some form? How is that here?
Yeah, we can just fine-tune the part of the pre-trained VAE model and then map the new data to the reference latent space. We don't need to retrain all the network structure. But if the user wants to retrain, it's still okay. But actually, they don't need to do this.